Good morning, everybody. Thanks for joining us for another episode of CRISPR Office Hours. We're going to give folks about 2 minutes to get logged on here before we get started. Look forward to our office hour session this week. All right, let's get started here. As always, I want to say thank you for joining us. Happy Thursday and welcome to Season 3, Episode 4 of CRISPR Office Hours. As always, for the past few months as we've been doing CRISPR Office Hours, we created this as a place for the scientific community to get together, talk about the challenges they're experiencing, and how we can collectively tackle these challenges and get through it together. And the various panelists that we've been privileged to have talked about different types of research that they've conducted, as well as how their labs are handling these turbulent times and the approaches they've taken to ensure that everyone is successful, showing empathy, and coming out of this better than we went into it. So as stated, our goal is to create a community, make sure there's a lot of interaction during office hours, and please send in your questions via chat. The more questions we have, the more engagement we have, the more value it is for everybody. And a few housekeeping items, please mute yourself and send in any questions via the chat. If you want to look at other office hours, these will be on YouTube from previous sessions, and then this also will be recorded and hosted on YouTube as well afterwards for your viewing pleasure. With that, excited to host another session of CRISPR Office Hours with my co-host, Kevin Holden.
Hey, Aditya. Welcome everyone to CRISPR Office Hours. I'm your co-host, Kevin Holden. I'm head of science at Synthego. The title for our episode this week is a combination B and T cell approach to vaccination against SARS-CoV-2. So our mantra over the past few months here has been to keep calm and carry on. And our version of it here at Synthego is to keep calm and CRISPR on. So please, everyone, stick around until the end of today's episode, where we'll tell you how to get a special edition Keep Calm and CRISPR On t-shirt. Our producer, Bobby, tells me that those t-shirts should be going out really soon.
So without further ado, Kevin and myself are very excited to have Dennis Hartigan-O'Connor, and welcome to CRISPR Office Hours. Dennis
Thanks.
is a MD, Ph.D. Professor and research scientist from UC Davis School of Medicine and works at the California National Primate Research Center in Davis. Welcome, Dennis.
Thank you.
Thanks for being here. So as we always do on CRISPR Office Hours, we have a little tradition. We like to go through and have a poll question and have folks chime in on their views on the poll. So this week's poll question we want to ask folks is, what activity what summer activity are you most looking forward to getting back to? And chime in with your response in the chat. One, social distancing pool party, short season baseball game, BBQs. Goal is to keep the sauce off your mask. Summer movies, make sure you watch it with the mask. 6 feet apart beach day, or outdoor happy hour where you lower your mask for sips. You want to chime in with what you do, Kevin? Start off with you.
I think the thing I'm most looking forward to is probably going to the beach. I know you can go to the beach in some places now and you can kind of keep apart, but just to kind of have like a nice relaxing day at the beach and take a nap. Looking forward to that.
Nice. What about yourself, Dennis?
Well, if I have to choose between these, I'd say choose happy hour, I think. But what I really like to do in the summer is make fresh jam from our East Bay plums here. Got a lot of plum trees in the neighborhood, so we'll be able to do that without the mask.
Oh, nice. Yeah, plums are delicious. I just had some Indian version of plum pickle.
Oh, okay. Maybe I should try to make that this year.
Yeah. Looks like a lot of folks are saying either a 6 feet apart beach day or outdoor happy hour. Looks like sun and engaging with people is the big theme for most of our CRISPR Office Hour attendees.
Yeah, I think I want to change my answer and just combine the happy hour with the beach. That sounds fun.
I think I'm going to go with you there too, Kevin.
Yeah.
Beach happy hour, social distancing. Cool. So to get started here, we've been going through this model and let Kevin kick it off.
Yeah, we've been working along this model of how scientific research may evolve and play out during COVID-19. And now we're starting to see the economy open up. And we're seeing this transition of people like going back into the workplace, back into research labs, into shopping centers and so forth. And hopefully we're all wearing masks, but is that really a good long-term strategy? So today we're going to talk to a scientist who's working on a more practical long-term solution to a viral pandemic, which is a vaccine. So let's get started. All right, so Dennis, thank you so much for taking the time to join us today. Before you talk about your vaccine strategy against SARS-CoV-2, can you tell us a little bit about your background and your lab?
Sure. Yeah, I like a lot of scientists working on SARS vaccines, come from a background in the HIV world. And because of that interest, I have done a lot of my work at the California National Primate Research Center at UC Davis. And when this pandemic erupted, UC Davis was one of the hospitals to treat one of the earliest Northern California patients. And as a result, the Primate Center got involved very early in modeling the disease and trying to understand how we could treat it. So I was kind of yanked from that pre-COVID world into COVID-2 vaccination. But, some of the same principles we believe and think apply here as an HIV.
What, maybe you can tell us a little bit about what makes UC Davis such a unique place to do this work. I'm a little partial, of course, because I'm a UC Davis alum for my PhD in microbiology, and I work next door to the Primate Research Center. So maybe tell us what you feel makes UC Davis kind of a unique place to do this work.
Well, UC Davis's traditional strength is in animal modeling. I mean, in the biomedical arena. And so when you have a condition like this, especially one that's pandemic, the urgent need is to find an animal model in which we can learn something about how the disease works, how to treat it, and how to prevent it. And actually, that is another way that HIV and SARS-CoV-2 are similar, and that the two of them are poorly modeled in mice. And there are some small animal species that are infectable with SARS-CoV-2, but essentially the best place to do this work is in an animal model that's naturally susceptible and in which we have a lot of tools for understanding the immune response, for understanding the virology and the immunology of the infection. And then, like I say, there's this happy coincidence for the progress of the research that UC Davis Health System was one of the earliest to treat a Northern California patient, and that we've had very close collaboration with our colleagues who spend most of their time in the hospital. And so it's really been a perfect proving ground, along with some other primate centers across the country, in which to do this kind of research. And in fact, it's one of the few, the total number of places in which you can study SARS-CoV-2 adequately is not so high across the country.
Can you tell us also who, before we get into your strategy and the work you're doing, can you tell us who you've been collaborating with? If you've been working with other groups, you mentioned some other places.
Yeah, so one of the unique things about SARS-CoV-2 is the level of national response, and especially the so-called public-private partnership, or active. And so, the major research efforts, or the largest research efforts, are being funded and done in cooperation with the NIH. And so, really, for the most part, most of your listeners will know that in most phases of life and under non-pandemic conditions, NIH is mostly focused on funding investigator-initiated research. In this case, it's different because we have a number of nationally prioritized strategies that NIH is working to develop. So we're working closely with NIH and with Active to prioritize the research that needs to get done. My lab and the Primate Center in particular have had a lot of key and important interactions with Vitalant. This is a blood supply company that's developed a lot of tools needed to understand antibody responses to the virus, both in people and in monkeys. And they've been a key partner in our studies in the monkeys. UCSF is, a traditional source of many great scientific connections at Davis, and a partner in a lot of the work we're doing and in our vaccine approaches. And we've also been working with Scripps on different versions of immunogens that might be used to prevent SARS-CoV-2 infection.
Cool. So let's hear a little bit about the strategy you've been developing, maybe some of the immunology behind it. I know your title suggests you're considering both B and T cell mediated stimulation. So maybe as we go through, you can tell us if one is going to be more appropriate than the other for SARS-CoV-2. So,
I think what's known at this point is that antibodies do work against the virus if you have enough antibodies, but that level of protection, the level of antibodies that might be needed is probably more than needed for certain other easier vaccine targets, and certainly is going to be most easily achieved in younger individuals. But the good news about SARS-CoV-2, in contrast to HIV, is that the antibodies work. That's something that isn't always the case. And in this condition, we at least know that this strategy of exposing people to the virus and allowing them to make antibodies that work in sort of the simplest way you can imagine to bind up the virus and prevent it from doing its thing is effective. So I just have a very quick example to show that what's achievable with SARS-CoV-2, if you can go to the next slide, is that monkeys in which antibodies are generated are also capable of inhibiting replication of the virus. So what we have here on the right is a number of vaccinees that have very nice antibody responses, and all of those vaccinees are able to prevent replication of the live virus. So this seems sort of obvious, I imagine, to non-immunologists, but actually this is not always the case. So at least what we have here is a correlative protection and a viable strategy. If you go to the next slide, one of the issues is, okay, it's a viable strategy in the sense that we know antibodies can inhibit the virus, but how much antibody do we need? And this is something that's beginning to be understood through the animal modeling studies being done, not just at UC Davis, but at many other primate centers as well. And this shows a result from Dan Brook's lab that was published in Science recently, which is really crucially important in the field, that begins to define how much antibody you need to protect the lungs.
Excuse me, can we have somebody mute, please? Oh, thank you.
Yeah, and so this is the kind of information that only an animal model can give you, and it gives you a target to hit for, and what's shown really is that the animals on the left that were completely protected, at least in their lungs, have met that target level that we'd like to meet with other vaccines.
If your vaccine isn't producing enough antibody, what sort of problems can that lead to?
The concern with any respiratory infection, and especially an RNA virus caused infection, is the possibility of enhancement, which is a situation in which not having enough antibody turns out to be worse than having no antibody at all. And this is something that has been a concern from the beginning of the pandemic, that as we try to develop vaccines and we have sort of variable levels of success, is there a danger that an individual who's recovering perhaps and their antibody starting to wane, or an individual who has a poor antibody response could experience worse infection. And so far that hasn't been the case for the most part. The evidence that's out there says that this doesn't seem to happen as badly as it does with SARS-CoV-1. And so we are optimistic that this at least won't be an impediment to developing a vaccine, as it has been for other diseases, especially RSV and dengue virus. Because you can imagine, if you have to thread that needle, that developing the vaccine becomes much more difficult. The other problems that we know are going to happen, because they're the case with sort of every vaccine candidate, are problems of first durability in response. Most of the newer vaccine technologies that are being tested here are thought to elicit antibody responses that are going to peak and then decline fairly rapidly. And so with something like flu, where you have a well-defined seasonality and you can give the vaccine immediately before that season and match your response to a seasonal infection, then durability is a problem, but perhaps a more manageable problem. With SARS-CoV-2, which is looking to be perhaps less seasonal, we might want a response that's going to last for a year or 2 years or 5 years. We don't know if our current vaccine technologies can achieve that. And then age dependence. People who are either elderly or infirm tend to have antibody responses that are worse and that decline more rapidly. And so it's crucially important that we do something that actually isn't usually done, which is to test all of our vaccine candidates in older monkeys, and when we get to people, in older people, to find out if they're mounting the kind of response that they need to protect themselves.
Okay, so also I wanted to ask you, you mentioned the B and T cell approaches. Maybe you can walk us through a little bit about how each one of those is important in terms of the immune response that's elicited.
Yeah, so the sort of, what we're questioning is, well, what kind of antibody response is needed, and do we have the tools to meet that challenge? And the answer is that it's a continuum. We do have the tools to make an antibody response, and it's going to be persistent for some amount of time. But one of the answers to that problem of waning or responses and waning protection is to back up the antibody response, for example, with a T cell response. And we know that antibody responses can be protective, so we don't want to ignore that arm of the immune system. But it's also clear from studies, not only in most of the most infectious diseases, but also specifically in SARS-CoV-1 and in MERS, that CD4 positive T cell responses in the lung can be an important contributor to protection. And when you make a vaccine, you can sort of choose which mechanism you're most trying to tickle. Antibody responses are mounted most commonly to things that are produced extracellularly or injected proteins, whereas T cell responses can be specific for antigens that are produced inside the cell, and T cell responses are perhaps somewhat longer lasting. So you can choose a vaccine strategy that tilts you in one direction or the other. And so one of the approaches that we're pursuing is to sort of combine both approaches to make sure that we're delivering vaccines that are targeting both pathways at the same time.
We did have a question, just to interrupt you real quick, that came through the chat. Somebody did ask, back to your point about not having enough antibodies could actually be more dangerous than having no antibodies. Can you maybe retouch on that a little bit?
Yeah, there are some infections for which it's known to be true that that what can happen is that because cells can bind to antibodies, some antibodies, if they're of the wrong type or they're directed at the wrong part of the virus, can cause the virus to be taken up into cells and then to initiate a self-perpetuating infection. The key is that there's usually a set of antibodies that are the right antibodies that don't do this, that are neutralizing antibodies, that are directed at the right epitopes and present at high enough level. So the key as we evaluate these vaccines is to be alert to the possibility of enhancement and make sure that we check for it. And I should say that so far we haven't seen evidence for that in monkeys, and so we hope that won't be the case. But it is something that all vaccine makers and researchers are on the lookout for, because we want to make sure that that isn't the case with this infection.
So would it be, just kind of back to your point about the T cell responses, maybe they're mediated through the antigen presentation. So would it be that T cell responses are going to be more for maybe the NSPs and the proteins inside the virus that it's making inside the cell versus the spike protein, for example?
Yeah, exactly so. I mean, there's a range of different T cell responses, some of which help the antibody response. So in these cases, you may need a whiff of that kind of protection. But we're really talking about protecting the virus at two different phases. Antibodies work on a virus that's been secreted from a cell and is floating around in the extracellular space, and antibodies can bind to it and inhibit it. And there's a separate class of T cell responses that can inhibit the virus inside the cell. And so that would be sort of hitting the virus at two different places. And the ideal vaccine, I think, would combine both of those approaches. I think the first wave of vaccines are all targeted primarily a generation of antibodies, although there will inevitably be some level of T cell response. I'm thinking of a bit more of a conscious effort to provoke both arms of the immune system.
Cool. So maybe you can tell us a little bit about the B cell and the memory response you can get from that.
Yeah, so there's a lot of different strategies to achieve this. One that's been pursued very heavily in the HIV arena is to take an antigen that might be a monomer, which is to say that as on the surface of the virus, you just sort of have single copies of this protein floating around, and instead of delivering it as a monomer, to deliver it as a multimer. So if you go to the next animation, the primary need of a B cell to develop into an antibody producing cell, and specifically into a high affinity antibody producing cell, the primary need of the B cell is to get help from a T cell. And that help from T cells is very limited by the availability of antigen specific T cells. And so on the left here, you can see that there's actually a competition for which B cell has the best receptor to get the antigen in the first place, and then which B cell can receive help from a responding T cell. And one way to begin to mitigate that problem and create sort of more B cells that create more antibody, and again, this is a strategy that's been pursued very heavily in HIV, is to take an antigen and multimerize it, both so that the B cell is more able to internalize that multimerized antigen and more able to present the antigens that have been internalized and more able to get T cell help. So this is sort of a second generation of immunogens that we and others will be pursuing, in which has been pursued, as I say, in other areas, but the first round of vaccines haven't yet taken this approach. So I think everybody in the field is expecting that we're going to have vaccines that are partially effective at first, and that there's going to be a need for second or third generation technologies that provide better, longer lasting responses. And this is just one of those approaches you can take to doing that.
Kind of like more shots on goal, I guess.
Mm-hmm.
Yeah. All right, maybe if you want to move on to the next slide, we can talk a little bit about the T cell responses you're hoping to stimulate.
Yeah, so in the same way, as it is the case that B cell responses to a vaccine are limited compared to the responses that you can expect in a natural infection, T cell responses are limited in the same way. And there's actually a balance between two different kinds of cells. one kind is outlined in yellow here and shown at the upper right. And these are stable memory T cells that are going to persist over a long period of time. And the other is these effector cells at the lower left. And it's really the former stable memory cells that are the ones that we want to live in a person for an indefinite period and protect them. And so on the other hand, it's these effector cells at the lower left that are sort of easiest to elicit, to follow, to assay in a short-term experiment in a monkey or even in a person. So we're very focused on finding ways to, and these two populations are actually traded off against one another. It's thought that cells that differentiate into effectors in certain cases are more likely to die than to go on and become stable memory cells. So we're focused on approaches that tweak this pathway to increase that sort of flux, that development of cells into those that are going to persist over the very long term in the animal or in the person. And we're again leveraging technologies and insights that come from HIV vaccine research to tweak these pathways.
Great. Can you maybe move on to the next slide here? Let's see.
And the next after this, I think.
Yeah. There we go.
Yeah. So fundamentally, the vaccine regimen that we are exploring in animals and eventually in the clinic is to combine these two approaches. And here are the two cartoons in the middle just show antibody responses and antibodies and T cells. And the approach is to prime with one adenoviral vector and to boost with another, and to use vectors that carry these novel either multimerized antigens, so in the top half of these circles, or with these memory promoting T cell antigens, which is shown in the bottom part of the circle. Okay, next slide. And what we know so far is that these approaches are producing responses that we do expect to be protective in people. I mentioned earlier that there's a threshold below which we think that people won't be completely protected. By using some of the approaches that I've mentioned, we're seeing responses in the monkeys that more than meet, that are more than adequate. But this slide, I think, illustrates some of the major issues in SARS-CoV-2 research, vaccine research, which is when we do an animal study, we often test protection in younger animals at the peak of the response. So here, for example, these animals, which are a relatively younger set, reached a peak on April 27th. And this is frequently a time period at which many folks would test for efficacy of their vaccines in these younger recipients. What we need to do instead is to test the longevity of these responses and the continued protection at periods that are, 1, 3, 6 months, even a year as we get farther in the pandemic after vaccination. And so obviously to do those studies, we need to vaccinate a lot of animals now and see how their responses evolve over time. But this is sort of the fundamental problem that our strategies are focused on, which is how to maintain all of the animals above this dotted line.
A question I had actually was, given that we're also moving into seasonal flu season, like you mentioned flu earlier, are you interested in looking at maybe how co-infection might affect an animal as well?
Yeah, I would say that right now we don't have the, right now I would say that the capacity for doing coronavirus studies in non-human primates is such that we don't have the capacity to do both. Right now there's a lot of vaccine candidates from both large companies, individual investigators that all need to get through the pipeline of being tested early and later. One of the nice things about testing the longevity of a vaccine response is that we can vaccinate those animals today and we can test them in 3 months, and they don't take up capacity for infection with SARS-CoV-2, which is limited across the country. Studies like you're talking about are of huge importance, but probably secondary to some of the more acute needs to understand the basic parameters of what's required to protect a person from SARS-CoV-2. So I think that's a second round of experiments. It's going to start in the, the fall or the winter of next year.
I guess also kind of based on this timeline we're looking at here, when might you predict that you have enough time, I guess, to say that, hey, we've developed memory or long-term protection in these animals, and that you could say, hey, now we're going to want to move into a human trial?
I think these responses tend to follow a fairly stereotypical pattern of a peak and then a decline that's more rapid initially, and then actually fairly fair stability over a long period thereafter. And so I think that as we use these vaccine candidates more and more, we will get to know the shape of that curve, and it'll be clear when the long-term response is going to be maintained. But I think you have to follow the animals for at least a few months to get a sense for what the kinetics and the dynamics of the response are going to be, and how much you're learning on a daily or monthly basis.
I'm sure there's one question that folks have on their mind. Sorry, I was muted there. Is what are your thoughts about the possibility and timeline of a vaccine being developed and available for the masses?
I think that people who develop vaccines know that there's a continuum. All right, there's a continuum from a vaccine that doesn't provide any protection to a vaccine that protects a certain fraction of people who are vaccinated, say 10%, to one that's really useful, that's protecting 60%, to one that's uniformly protective, which would be similar to our, measles vaccines or our polio vaccines at the upper end. And all of these vaccines then have a time course on top of it. So you're going to have vaccines that are 60% protective the day after they're given, and that the protection declines over time to a certain level. So my feeling is that we'll have a vaccine for sure, and it will be given to a lot of people for sure. And it's also probably going to provide protection that's not 100%, that's imperfect, and that lasts for only a limited period. So I think we're looking at a series of coronavirus vaccines, of which the later ones are going to be better than the earlier ones. I think that's inevitable with development of any technology. But I don't think it's likely that we're going to have no working vaccine at all. I think that's an unlikely outcome. I think what we'll have is a suboptimal first generation vaccine that needs improvement.
Kind of going back to the some of the immunology a little bit. You talked about both T cell responses and B cell responses. I guess some of us have been hearing a little bit about this kind of like overreactive, like T helper response. Like, is that a concern when you're developing a vaccine strategy that you could basically activate this kind of like overreaction type response?
Absolutely, yeah, and that right there is the key reason for doing animal modeling and safety studies in animals before going into people. It's really important that people understand that this is the reason we do work in animals, is because we don't know all of the implications of these strategies. We do think we have a pretty good model for the infection in not just in monkeys, but also in certain types of hamsters and even in some transgenic mice that can answer that question before we get to people. And that's one of the reasons that there's a lot of urgency to continuing with the work in animals, even as we start testing the vaccines in people. Because the one thing we can do in animals that we're not yet ready to do in people is we can challenge them with the virus and see if they do better or worse.
Can you, speaking of that, can you, obviously there are some already ongoing vaccine trials, I guess, and we've heard about some of these in the media, and the vaccines developed by Moderna and like Oxford University. Can you maybe talk broadly about what you've heard, at least about how the success of how these trials may be going, or what the kind of hope is for them? Do you feel like they're maybe just going to give like a bit of short-term immunity?
I think the evidence is that they're immunogenic, that they work in the sense that they make neutralizing antibodies. And I should say, evidence is not available for all of them. There are some candidates that have been tested in animals, and some that have been tested in animals, but the results haven't been published. And in many cases, the assays that were used are different between studies. So I think what we can say for sure is that we have a number of candidates that are immunogenic, and that they make the kind of immune responses that are needed for protection. I also think we haven't seen yet a candidate that produces responses that that are sufficiently intense, I think, to protect 100% of people. I think that's an unlikely outcome for the things that have been tested so far. But the only way to answer these questions is to get into humans. Humans and animals have different immune responses in magnitude and in dynamics. So what's been seen in animals is that they're immunogenic and they work well enough to provide some level of protection. But as far as determining how much that is or how long it's going to last, really we have to hope that the results of the human trials are similar or better to what's being seen in the animals.
So a few weeks ago, we had a collaborator of ours on from UCSF who's working on therapeutics. So and they're actually targeting human proteins, right? And part of the justification for that is that human proteins are less likely to mutate than viral proteins. And we had a question come in the chat here that asks, is there any data on the mutation rate of the virus which could complicate the availability of an effective vaccine in the long term?
All the data are that the mutation rate is not a problem. That's what we, every indication that we have is that that's the case. It's not, we don't have potentially enough vaccinated animals and people to know if the mutation rate could be higher in the presence of these inhibitory factors that obviously tend to select for resistant viruses. But the evidence that's available is that the mutation rate is low, and the mutations that have been found to be common don't affect the vaccine antigens that are the focus of most research. So, right at the moment, we're very alert to that possibility, but it's not turning out to be a huge concern yet.
I guess another quick question I had was, the antibodies, that we're starting to see more and more antibody data come out, and how might that inform, like, maybe the next generation of vaccines?
Well, the beautiful thing about some of the antibody data is we're learning a lot about individual antibodies and exactly what parts of the virus they attack, and that also gives us the ability to produce these individual antibodies in a factory and give exactly as much antibody as we want to give to an animal or eventually to a person, and find out how much of that antibody is protective. These kind of neutralizing studies that I've presented here give an inkling, but they're also measurements of the group properties of a large number of antibodies in serum. And as we get individual antibodies, there may be those with extraordinary activity that are extraordinarily effective even at low concentrations. So we're really learning about the precise characteristics that are needed and how much is needed to protect people.
Okay, I think we had some also questions from the audience that were submitted beforehand, but I know you'd obviously like to give some acknowledgements to your lab and your collaborators. Yeah,
I just want to say that it's been really inspiring. As I'm sure many of your panelists have commented on, to see the degree to which people have pulled together. We got into this work not only because it seemed obvious that some of our vaccine technologies would be applicable here, but also because, people in my lab and outside my lab at UC Davis stepped up and said that they wanted to use their skills in growing the virus under high containment, or in animal modeling, or in pathology, to study this urgent public health crisis. And it's the kind of situation which people are reaching out to you, asking to do work and get involved. So that's been really inspiring. And all these people listed here in my lab and my collaborators are people who have done that.
Cool. Yes, some fantastic names there I recognize from my time at Davis. Peter Barry, Liz Sparger, yeah, Danica, Chris Miller. So yeah, I will I put a little plug in and say if you're looking to do some great science, either as a grad student or postdoc, check out UC Davis, either Center for Comparative Medicine or the Primate Research Center. Some great translational science happening there.
For folks that don't know online, Kevin is a alum of UC Davis.
Yeah, I hit that already.
Mm-hmm.
All right, so we had some questions come in. Did you want to read those?
Yeah, sure. As part of our office hours, we asked for questions ahead of time, and we got some that we want to make sure to go through. Tim from Gilead, how likely is it that one of the many vaccines currently under development will work in preventing COVID-19 infections, and why?
Again, I think it's certain that one or more of the vaccines under development will work in preventing COVID-19 infections over the short term, and in people who mount an adequate response, right? Like one of the challenges of HIV vaccine development, speaking to somebody who's at Gilead, is that we don't know what response is actually required to be protective. In COVID-19, we do know that. And I think we know that several of these candidates will elicit that response. The real question is in how many people and over what time period, and will it work in the people who need it the most, including elderly people and those with comorbidities.
Okay, great. Got another question from Guy Andre from Toulouse. Are the genomics susceptibility or resistance to coronaviruses, and especially to SARS-CoV-2?
Yeah, this is not my area of expertise. I will just say that I know that there's a lot of interest in this. We are collaborating with people who are interested in asking the same question in non-human primates. So if monkeys to be infected with SARS-CoV-2, can we look at the genome of that animal and understand reasons for divergence in the course of disease? And people are undertaking similar studies in humans, but I don't know the details of those studies. It's not my primary area of focus.
Okay. Then we have another question from Maya from DePaul University. If CRISPR is to be used in the treatment of COVID-19, what steps would be taken to identify potential off-target mutations that may arise?
Maybe this is better for me to answer this one. So we actually, we've seen a couple of papers come out where people have used these CRISPR genome engineering approaches to try to target the virus in vivo using a nuclease that actually will specifically target and chew up RNA. So like Cas13 nuclease. So I think, there's a couple of papers out there already on that. Probably, I'm sure people are doing pretty deep due diligence to make sure there's no off-targeting with, any human transcripts. But, again, I think we're gonna have to wait until we see how those play out. And obviously, delivering those reagents basically into a patient can be challenging. So maybe we'll try to get a speaker come on in the next couple of weeks to talk about the strategy they're working on for that. So stay tuned. And it looks like someone also has asked about CRISPR for virus detection. And yeah, the link is here for our website, and we have some great information about speakers we've had in the past who are using CRISPR for diagnostics for COVID-19.
Great.
All right, so we're back to the model here. So Dennis, maybe do you feel like we're kind of in this is a model that a research group called the Lioness Group put together, and they interviewed over 2,000 scientists just to kind of get an idea of like how they think research is gonna happen. Obviously for scientists who are maybe taking time away from the lab right now, maybe as we're going to this kind of like new normal type situation. People are talking about a second wave. Is that really a real thing, or is it more in your eyes? Do you see like we're still in the first section here, and we're starting to see more infections because people are just kind of becoming relaxed in how they're protecting themselves?
Yeah, I mean, personally, I think it's semantics a little bit, but I don't see a big I don't see a distinction between these waves at all. To me, it's just a constant cycle of infections in which there's fuel for the fire, so the virus is going to go onward. So yeah, I would call this first wave, but I'm not sure it matters because there's a lot of, unfortunately, a lot of infections out there.
How,
Okay.
so as you go forward, one quick question, as you were looking at this model, I just want to ask, where is your lab and where do you think you guys are?
I think my lab's a little bit unique because we're working so much on the virus and in non-human primates, which are really part of the national response to this.
Yeah.
So, gosh, my lab's, I feel like it's doing about, 3 or 4 times as much work as we were ever doing before the pandemic, just because people are so engaged and there's so much demand and importance to move the work forward. But the one thing that intrudes on us frequently is the need to sort of keep our HIV work moving forward, which, even though we're still operating, unlike many labs, is inevitably taking a back seat. And I've been thinking frequently about the fact that this is going to have a major impact on all sorts of research foci. I mean, if it's having that degree of impact in my lab on areas that are less focused, then I think it's going to have a much bigger impact on labs that are going to be shut down for longer. And I think there's also a lot of impact from the travel ban, or not the travel ban, I misspoke, from just the reduced amount of travel needed to get new people into positions, new students and postdocs to the universities, new people on campus. I think it's going to have a sustained negative impact on research output in other areas. That maybe we need one good therapy and we need several good vaccines to really get back to normal.
Yeah, that's a great point. So one thing, as we have office hours every week, next week we actually will be off. But in lieu of that, we're actually really excited for all the attendees here, folks who've registered. We'll be sending out something special to your inboxes next Thursday. So keep an eye out for that. It's a little something that we've been working on at Synthego here that relates to COVID-19 and a few things we've done around it. But with that, Kevin, who do we have when we get back?
Yeah, so speaking of the model, we just went over the person that put that model together, Hamid Ghanadan, is going to join us again. So he joined us way back at the beginning of March when we just started the shelter in place and kind of introduced us to his idea. And so he'll be back with some fresh insights from new data they've collected and observations from the past few months. So it'll be interesting to see how accurate that model is. So again, that's going to be in 2 weeks. Because we're off next week. We have a short week for the Independence Day holiday. But as Dennis alluded to, definitely we can concur with that. As Synthego, we feel like we've actually been working even harder than we normally do pre-pandemic. So yeah, we'll look forward to having Hamid on in a couple weeks and kind of talk us through how he thinks things are going to pan out as people can get back into the lab and start to pick up important research, like Dennis said, that has kind of been taking a back seat. All right, and just to remind everyone, if you want to get one of our Keep Calm and CRISPR On t-shirts, and Dennis, we can send some of these to your lab if you want. I'm not sure if you guys are doing much CRISPR, but you can go to synthego.com/calm. So again, just want to thank Dennis Hartigan-O'Connor gracefully for appearing on CRISPR Office Hours this week. Thank you so much for taking time out of your busy schedule to tell us about your vaccine strategy and the work your lab's doing. And really, we wish you all the best of luck, and we really need it to work.
No problem. I enjoyed it. Thank you.
Thank you, Dennis. Appreciate it. And to everyone online, just remember you can get this on YouTube, on our YouTube channel, youtube.com/Synthego. We have previous CRISPR Office Hours also, and we do a blog recap. With that, we want to thank everybody for attending, and I'll look forward to seeing you guys in a few weeks. And keep an eye out next week in your inbox for something that we're going to send out. Thank you.
All right, great. Thank you, everyone. See you next time. Thanks, Dennis.